Western blotting. Today we are going to see about Western blotting. Tobin in 1979 developed the Western blotting or protein blotting to find out the newly encoded protein by transformed cell. So here in this technique for detecting specific proteins separated by electrophoresis with the use of labeled antibodies, so called since it has some similarities to southern blot test. Definition: the Western blot is analytical technique used to detect specific proteins given in a sample of tissue or homogenate or extract. Western blotting is a rapid and sensitive assay for detection and characterization of proteins. It is based on principle of immunochromatography where proteins are separated into polyacrylamide gel according to molecular weight. So here is the process where the tissue preparation is done. Next, gel electrophoresis. After that, the proteins are transferred according to their molecular weight. Then the blocking of the proteins according to the molecular weight. Detection with the help of radioimmune assay or labeled antibodies. And finally, the analysis. So here is the process of preparation of sample. The cells are lysed and centrifugation is done. The cytoplasmic proteins and nucleus proteins, they are separated according to their molecular weight. As per our interest, the sample is selected by micropipette and then it is used for further process. Next is SDS page where sodium dodecyl sulfate is used. With the help of this process, the proteins are separated according to their molecular weight. The sample is loaded in the wells as shown here, and one ladder cell is there which shows all the type of proteins according to their molecular weight, which we are going to compare for our target proteins. So this is the process by which the proteins is transferred from gel to nitrocellulose membrane. The membrane transfer, this process is used by applying positive and negative charged. Because of this, proteins are transferred from gel to blotting membrane. And after the protein is appeared on blotting membrane, then it is separated by using labeled antibodies. Finally, here it is the process how the proteins are transferred and labeled on exposed film with the help of labeled antibodies. These are the proteins from the cell which first we are transformed on SDS polyacrylamide gel, where we are separated them according to their molecular weight. After that, the labeled antibodies are placed, and finally interested proteins are separated. So here, after every step, these plates are washed. So unwanted or those who are not needed, such kind of proteins are removed. And finally, the interest proteins are separated out from the total cell proteins. Thank you.